Hello everyone, welcome to Get My Class. So let's solve this question. It says, which of the following is false for DNA polymerase? Option A, DNA polymerases are highly efficient enzymes as they catalyze the polymerization of a large number of nucleotides in a very short time. Option B says polymerization occurs in 3' to 5' direction from the start point to end point by DNA polymerase in E. coli. Option C says it completes the process of replication within 8 minutes in 18 minutes in E. coli. Option D is none of this. So let us understand the question. It says which of the following is false, or you can say incorrect, for DNA polymerase. So before we look for incorrect statement, first of all we need to understand what are these DNA polymerase. Okay, what is DNA polymerase? It is a type of enzyme. Yes, it is a type of enzyme. There are different variants of DNA polymerases that we have to remember. And the main function, as the name indicates, polymerase, that means it's an enzyme which brings about what? It brings about polymerization. And since it says DNA polymerase, that means it's bringing about polymerization of what? Polymerization of nucleotides. Okay, and what kind of nucleotides? It is bringing about the polymerization of deoxyribonucleotides. Okay, deoxyribonucleotides. That's why it is DNA polymerase. Now, yes, it is very efficient. Okay, it is very efficient. It can add number of nucleotides. You can say large number of nucleotides per second. Large number of nucleotides, nucleotides per second. Per second. Okay. So they function very, very fast, and they are very helpful during one is during DNA replication because they are helpful in the synthesis of the DNA as well as during DNA repair mechanisms. Whenever there is a breakage or whenever there is loss of a piece of the DNA, these enzymes will help in repairing as well. Not all, but again, there are different types of DNA polymerases. Now, the direction in which they work is 5' to 3'. Okay? So they add 5' to 3', the nucleotides in the 5' to 3' direction. Okay, so that is again. And one more important point about DNA polymerase is that these cannot directly start synthesizing. They need 3' hydroxy end to add nucleotides. They cannot just start synthesizing the nucleotides. Okay? So they cannot just start synthesizing or the new DNA. They need the presence of 3' hydroxy end. And this is provided by the RNA primer. So let me make you understand. Suppose this is the DNA template. This is the DNA template. For it to start, and this is 3' to 5'. The template is 3' to 5', whereas the polymerase will synthesize in 5' to 3' direction. So it cannot directly add nucleotides here. So it needs a primer first. It needs a primer, that is RNA primer. What is this? This is RNA primer. Okay, the function of RNA primer is to provide the free 3' hydroxy end. Okay, 3' OH group it will provide. And it will again 5' to 3' direction. So when it provides 3' free 3' hydroxyl end, now the DNA polymerase will come into picture, and now it will start adding the nucleotides here. So it cannot directly start the synthesis of the DNA without the help of the RNA primer. Okay. So once it is synthesizing, later this RNA primer will be removed and it will be replaced by deoxyribonucleotides. Okay. So let us read now. The option A says DNA polymerases. One second. The option A says DNA polymerases are highly efficient enzymes as they catalyze the polymerization of a large number of nucleotides in a very short time. That is absolutely right. Option B says polymerization occurs in 3' to 5', which is incorrect because, as I told you, it occurs in 5' to 3' direction. Okay, one thing. And it says direction from start point to end point by DNA polymerase, which is incorrect because start point, it needs the presence of RNA primer. It cannot directly add. And this RNA primer is added by RNA primase enzyme. Or you can say, yeah, RNA primase. Okay. Whereas DNA polymerase will start from here. Okay. So this is incorrect statement. But that should be our answer because we are looking for false statement. But let us see others. Option C says it completes the process of replication within 18 minutes in E. coli. That is absolutely fine because the generation time for E. coli is about 20 minutes. That is the cell division time, 20 minutes. But the replication of the DNA takes up within 18 minutes. That is absolutely right. That means the statement is right, but that is not an answer. Option D says none of this, which is incorrect because we have an answer that is option B. So option B is the false statement. This should be our correct answer. So let us conclude the answer, which is the false answer, or we can say false statement. That is option B. I hope you understood the explanation. Thank you, everyone.